Hello guys. In the last class, we discussed about the RIA, that is the radioimmunoassay. Today we will discuss about the ELISA, that is the enzyme-linked immunosorbent assay. In the case of RIA, in the last class, when we talked about the RIA, we studied that in the case of RIA, a radioisotope is attached to the enzyme. A radioisotope is attached to the antibody, and the radiation emitted from this radioisotope, this radiation emitted by this radioisotope gives the idea of presence of compound in the sample. Okay, that was the basic idea. But this radiation may prove to be hazardous for the researchers. It is expensive as well as has some negative effect on the researchers or in the laboratory. Okay, to move to a more safer strategy or technique, that leads to the evolution of development of this ELISA technique. In ELISA technique, instead of radioisotope, an enzyme is present or tagged with the antibodies during the estimation. Okay, and this enzyme which is attached to the antibody is known as reporter enzyme, because this reporter enzyme only is responsible for giving the idea of presence of a particular type of immunogen in the sample. Okay, antibody or antigen. Okay, how it happens? That means that this enzyme converts a substrate into a colored product. It converts into a colored product, and the development of colored product gives the idea of presence of a particular substance or qualitative test of the sample. And also beside that, the intensity of the color gives the amount of that particular sample or that particular molecule or that particular antigen or antibody present in the sample. Okay, so this development of the color gives the qualitative analysis test, and the intensity of the developed color gives the idea about the quantity of the that antigen or antibodies, or it gives intensity of the color is the basis of quantitative analysis. Okay, so this is the difference between the RIA and ELISA. In the case of ELISA, a enzyme is attached to the antibody, but in the case of RIA, this in place of enzyme, a radioisotope is present, and in the case of ELISA, development of color is the basis of the assay, and in the case of RIA, the counting of the emission is the basis of analysis of our assay. Okay, so that means in enzyme-linked immunosorbent assay or ELISA, antibody is attached to an enzyme. That enzyme is known as reporter enzyme because it reports about the presence of a particular antigen or antibody. That is why it is known as reporter enzyme. And the reporter enzyme converts its substrate into a colored compound. Generally, colored compound is known as chromogenic because sometimes we develop a color. Okay, and the intensity of the color is directly proportional to the concentration of that molecule, that antigen or antibody. Okay, the whole process. In the process, the one antibody is attached to a solid base, and on that attached antibody, the experiment is carried out. That antibody may be attached to a polystyrene surface, or generally it is coated on a specific plate called ELISA plate. It contains several wells, okay, and that wells is coated with the antibodies, okay. So this whole experiment is carried out in a specific plate, a special plate called ELISA plate, okay. So there are two types of one more thing. Okay, the reporter enzyme, generally most commonly used reporter enzymes used in the ELISA are phosphatase peroxidase or alkaline phosphatase or beta galactosidase. These are the enzymes used as reporter enzyme, okay. In the out of these, most commonly used is phosphatase peroxidase. It is a peroxidase, that means it converts H2O2 into H2O, okay, that reduces H2O2, and for that purpose a electron donor is required, which that electron donor, after donating electron to H2O2, gets oxidized to form color product. So the substrate is basically here the substrate is H2O2 for in the case of peroxidase, but there should be one more compound needed. One more compound is needed. That compound is the electron donor, which donates the electron to get itself oxidized and form colored compound. Most commonly used electron donor for horseradish peroxidase or tetramethylbenzene or orthophenyl dihydrochloride. These compounds are generally used for when the horseradish peroxidase is used as reporter enzyme, which convert H2O2 in H2O into H2O, and for that purpose these two compounds are generally used as electron donor, and after donating electron these compounds get oxidized and form colored compound. That in the case of tetramethylbenzene, a blue color compound product is developed, whereas in the case of phenylene diamine dihydrochloride OPD, yellow color develops, and this development of color give the idea about the molecules present in the sample, either any antigen or any antibodies. Okay, so there are two types of ELISA. Basically, there are two types of ELISA. Okay, indirect ELISA and double antibody ELISA. That is double antibody sandwich ELISA. That is the DAS. Okay, but indirect ELISA is also a sandwich ELISA, but here in case of double antibodies, triple antibodies are used. That is why it is also known as triple antibody sandwich ELISA. Okay? It is called indirect ELISA because by this method one can detect or diagnose the presence of an antigen by diagnosing the presence of antibodies in the blood formed in response of the presence of that antigen. If antigen is present in the body, that antigen will result in formation of antibodies in the body by the immune system, and presence of antibodies for that particular antigen will give the idea about the infection. If that antibodies which is formed in response to the antigen is present in the body and we detect or evaluate that antibody, presence of antibody. If antibody is present, we can say that the antigen is present. That means the diagnosis of the antigen is carried out by diagnosis of antibodies present in the patient's body or anybody, human body, okay, or animal body, okay. So that we can diagnose the presence of an antigen by diagnosing, detecting the presence of antibodies in the body formed as in the response of infection. Okay. So by suppose that this process is used to diagnose the infection of hepatitis B virus or AIDS, HIV virus. Okay, HIV virus or hepatitis B virus. By detecting the antibodies of these two, either HIV or hepatitis B virus, by detecting the presence of their antibodies in the body, human body, we can diagnose indirectly the presence or absence of that antigen or that virus, either HIV virus, or we do not diagnose directly the presence of virus in the body, but rather we diagnose the presence of, we evaluate the presence of their antibodies in the human body. If antibody is present in the body, that means infection is present or that particular patient is infected with HIV or AIDS, whatsoever the diagnosis is going on for. Okay. So we discussed about the indirect ELISA. It is used for detection of, it is an indirect detection of for HIV or for hepatitis B virus. Okay? So we can first we detect the antibodies. Detect the antibodies of HIV or HBV. So by detecting the antibody, we can diagnose the infection of either HIV or HBV. That is why it is known as indirect ELISA. That is because it is an indirect detection, a process of indirect detection of the infection of either HIV or HBV. So, what is the process? We can discuss it by this diagram. Here, suppose this is the surface. Suppose this is the surface of the well where antibodies is coated. Okay, these antibodies are non-specific and it is for any particular virus, for example HIV or HBV. So these are the antibodies. First of all, these antibodies are coated to this solid surface of the well. Suppose this is the solid surface of the well, and these antibodies, when added to the well, they get attached by their FC segment to the surface. Okay. One more thing is that these are antigen attached to these portions, but these portions of the well is exposed where unwanted protein can get bind and hampering the procedure of the test to prevent that possibility, some neutral compounds, some neutral molecules are allowed to bind to this exposed surface. So these are the neutral molecules which are allowed to bind to this free exposed surface in order to prevent binding of any undesired molecule present in the sample. So these neutral molecules, non-reactive molecules, may be either bovine albumin or it may be casein. Okay, so these are used to coat the free surface to prevent any binding of any undesired molecule during the procedure. In the next step, the antigenic part, the only antigenic part like the envelope of HIV, okay, spikes of HIV is used, okay, and that which antigenic part, that means which bind with the antibodies, only that particle is required, and the whole virus is never used, because the using a live virus or whole virus will give a hazardous effect, will give a very lethal effect. Maybe if infection happens to the researchers, that will get sick. That researcher or that technician itself will get sick or infected with that virus. So that proved to be very lethal and very undesirable. So that is not done. The process is carried out. What is done? Only the antigenic part of that virus is used. Then in the next step binds to the antibody coated to the surface. For that purpose, a coat or envelope part of the antigen is used. In the case of HIV, antigen envelope part is used, and in the case of HBV, coat of the virus is used. Okay, so suppose this is the antigenic part of the virus. This is added to the veil, and this antigenic part will bind to the FAB region of the antibodies attached to this veil. So suppose this is the FAB, this will be the FAB antigen binding region. Okay, so to this portion of the antibody, this antigenic determinant will bind. This is the first case. In the next step, the sample is added. The sample containing the antibody. That means what will be the sample? The sample will be serum of the patient, blood of the patient. Okay, that sample serum is added, or that sample of blood portion is added to the veil. If the serum contains if that patient is infected with HIV or HBV or particular virus, the antibody of that virus will be present in its body, and as well in the sample as well. Okay, that antibody will obviously bind to the antigenic part by its FV region. So here a sandwich is found, where this is the antibody, this is the antigenic determinant, a part of the antigen, and this is the specific antibody specific to this antigenic determinant. That means if it is the HIV antigenic determinant, this antibody specific to the HIV will bind to this antigenic determinant, and this antigen will become sandwiched between two antibodies. One antibody which is specific to it, okay, which is present in the sample and other one which is coated on the ELISA plate. In the next case, a secondary antibody containing the reporter enzyme is added to the well, and this secondary antibody is anti-antibody. This anti-antibody, which is labeled with reporter enzyme, will bind through its FAB region to the FC region of the specific antibody. Okay, so this will bind here. This is the FAB region and this is the FC region. This is the FC region of the specific antibody and this is the FAB region. So that secondary antibody or the antibody containing the reporter enzyme will bind by its FAB region to the FC region of the specific antibody present in the sample. So that will form, the whole structure will become like this, where the antibody, secondary antibody containing the enzyme is attached to the FC region of the specific antibody, and this antibody will get attached to the antigenic determinant of that particular virus. Okay, and this antigen will be attached to the antibodies coated to the wall. What type of antibody are used, we will discuss in the next class when we discuss the double antigen sandwich ELISA. Okay, the whole structure will form like this. In the next step, the substrate will be added. Substrate is added, and that substrate will be converted by this enzyme into a colored product. Presence of colored product. In the each step, washing is carried out. Okay, antigen is added. This will get attached to it, and then it will be the well will be washed is washed, so that the antibody or antigen which are not bound to the antibodies, coated antibodies, will be washed off. In the next step, sample is added, and when the sample is added, they will be incubated. The whole process or the whole ELISA plate will be incubated. Apparatus will be incubated for a sufficient period of time, and after that the well will be washed again, so that the rest of the enzyme antibodies which is not desired specific, which is not attached to this, will be washed off. And the next case, in the next step, the reporter containing antibodies is added, and that will bind to it. Okay, again washed, and after that the substrate is added, color product is formed, and presence of color product will give the idea about the antibodies, because to these antibodies only these antibodies are attached, and which form color product. Had it not been present, this would have not been attached to this antibody, and during washing they all will be washed off and no color will be developed. So that presence of color product indicate that this antibody is present in the well, to which this antibody containing the reporter enzyme got attached and is not washed off during the washing period. Okay, and produce the color. So this production of color give the qualitative analysis of the antibodies, and the intensity of color is measured Intensity of color is measured by the electrophotometer. Okay? Electrophotometer, there is a technique for, there is the apparatus which can give the measurement of intensity of developed color, and by the formula we can estimate or calculate the concentration of the antibodies present in the sample. Okay, so this is the process of indirect ELISA, by which we can detect the antigen in antigen infection of a particular antigen in the patient by estimating the antibody specific to that antigen in the body of the patient. Okay, thank you very much.